My name is Geeta Galonska and I'm the head TA for the biotech program here at NSLC. Today they're doing gel electrophoresis. Yesterday they took DNA samples and used restriction enzymes to cut them at specific places, which they learned about in lecture. And today they're running them on a gel to separate them and see the actual DNA bands that formed. You can learn about these things in a classroom for a long time. The core of what happens in biotech and companies and research and all these things are the actual products that are so small and so complex that seeing them on a large scale, seeing them actually being run in these experiments, prepares them in such a good way for what they'll see in school and their careers in the future.
I'm Bernice and I'm from Mountain House, California.
My name is Sophia, I'm from the Dominican Republic.
My name is Madeline, I'm from Holyoke, Massachusetts.
Today we learned about the process of making recombinant DNA, and we learned about what we're doing in the lab right now and what we're carrying out in that experiment in depth. So we were able to understand what we were doing yesterday at the molecular level and understand what we were going to do later in the lab, which is putting blue dye into the DNA with enzymes, which will now separate the DNA into fragments, and we will be able to see the different sizes of DNA in the gel.
In lecture, we learned about cutting DNA and repasting it in the process, and we also learned about electrophoresis and how to do it. I've run an electrophoresis before, but I've never gotten to pour the gel, so I thought that was pretty cool.
Well, I've actually run a gel before, so the pouring of the gel was familiar, but some of the stuff we learned in the lecture, like looking at DNA at the atomic level and the names of the bonds, especially the ones that keep the sides of the DNA together.
Recently I read a book about molecular biology, so these are all things that I've kind of read about, but getting to actually see them in lecture and getting them explained in depth and at a deeper level is great because I get to understand a topic better that I already knew about. And the fact that I get to do it in the lab and do hands-on activities with it, understand how it works and see it myself is really a great experience, and I've enjoyed it.
I'm interested because I think it's an interesting idea that you can alter things that are already alive to make life better. The aspect of biotechnology I'm interested is biomedical engineering, because I think there's a lot that you can do to make medicine more efficient and it could help more people.
I'm interested in particular in working with biotechnology that would help marine life, because there's awareness about how inhospitable the oceans are progressively becoming as planetary conditions are worsening, so they might need intervention by humans to make sure that their species doesn't go extinct completely and unbalance the whole ecosystem.
Biotechnology is all about understanding biological processes and understanding living things and then altering them to make them useful for humans or for nature or animals. I love that you need to understand the topic on a deeper level, and in order to use that, you need to be able to understand it, and I really just want to learn how everything around me works and just be able to see things and get what they are and how they're working.